So now we'll take a look into microbial growth. When microbes are provided with the nutrients and all the required environmental factors, then they'll become metabolically active and grow. Growth takes place on two different levels. Cells can either synthesize new cell components and increase in size, or the number of cells in the population increases. The way we get the number of cells in the population to increase is through the process of binary fission. By far, most bacteria that have ever been studied reproduce by binary fission. Binary literally means one cell becomes two. However, recently we've discovered that some cells produce multiple offspring within their cytoplasm and then split open to release multiple new bacteria. This is a new idea, so culturally we really think of bacterial growth as binary fission. It's an asexual process. As you can see here, we have a young cell, and that young cell is replicated, and that young cell's chromosome is replicated. After chromosome replication, a protein band forms down the center of the cell, and then that septum forms through production of more cell wall. Eventually, the cell splits into two. So simply, we have replication of the bacterial chromosome and then division of the cell. Because the generation time in bacteria is so fast, within 24 hours, one cell can result in 5,100 tons of cells, or 10 to the 21 cells. So the rate is huge. However, it's limited by the environmental factors and nutrients available. Let's take a quick look at an animation of binary fission.
Bacterial cells generally increase in numbers by the process of binary fission. The cell increases in length while doubling all of its parts, then forms a cross septum and divides to become two cells. Two cells become four. Those four become 8, and so on. This pattern of increase results in exponential growth of the population. The time required for the number of cells in the population to double is called the generation time. Depending on the medium, temperature, and other conditions, some bacteria can double in numbers every 30 minutes. Other bacteria have generation times of several hours or even days. During growth of the cell, streptococci and some other gram-positive cocci synthesize new cell walls in the septal region. Rod-shaped bacteria such as Escherichia coli, Salmonella, and Bacillus species synthesize new cell walls over the entire surface of the cell.
Generation time is perhaps the biggest influence on the rate of population growth. Generation or doubling time is the time that's required for a complete fission cycle. So how long does it take for a cell to produce two of itself? In each fission cycle, or each generation, we increase the population by a factor of 2. As you can see here, 1 results in 2, and 2 results in 4, so on and so forth. So in essence, we can use the generation time as a measure of the growth rate of an organism. The average generation time for bacteria is 30 to 60 minutes under optimum conditions, but can be as short as 10 to 12 minutes. When we see this pattern of doubling, it's considered exponential growth. Now, we graph the data from growing bacterial populations by plotting the number of cells as a function of time. As you can see in the graph down here, if we plot this number logarithmically, it ends up being a straight line, as you see across the center. However, if we plot it arithmetically, we have a constant curve shape. This is where we come up with the term growth curve. To calculate the size of a population over time, we use this formula here, where n sub f is the total number of cells in the population at some point in the growth phase. It doesn't matter which, you'll see. Ni is the starting number, where little n, this is not a capital, denotes the generation number. So let's take a look and put an example to this. Let's consider an example of Staphylococcus aureus, and we'll calculate how many cells n sub f will be present in an egg sandwich after it sits in a warm car for 4 hours. you probably won't leave an egg sandwich ever again. We'll assume that Ni, the initial number of cells, is 10. Those are how many cells of Staphylococcus aureus we accidentally ended up putting in the sandwich while it's being prepared. Then we're going to derive N. So we need to divide the number of hours, 4 hours, that we left the sandwich in the car, which is 240 minutes, by the generation time. Let's use an average of 20 minutes for this particular bacteria. So when we divide 240 by 20 minutes, we end up with the number 12. So N is 12 generations. So 2 to the N is equal to 2 to the 12, because remember, little n is the number of generations that we have. When we plug 2 to the exponent 12 into a calculator, we find out that we have this huge number of 4,096 bacteria. So now we can plug that into our equation. N to the F equals N to the I, which was 10, times 2 to the number of generations, which we said is 2 to the 12. And we end up with 40,960 bacteria in the sandwich after leaving it in the car for 4 hours. Now you can do one of two things. Make sure that the preparation of your sandwich is absolutely aseptic, or don't leave it in the car for 4 hours. Let's have a quick concept check. If you have 100 bacteria with a doubling time of 20 minutes in media, how many cells would there be in 2 hours under optimal growth conditions? So here we go. Basically, we're doing the same calculation. The equation that we're considering is N to the F, the final number, equals the initial number times 2 to the exponent how many generations. So Ni in this case equals 100. NF is our question mark. What is little n? Well, little n, we have a doubling time of 20, and we're growing for 2 hours. 2 hours is 120 minutes. So we have 120 minutes. We're going to divide it by 20 minutes per generation time. So n is going to equal 6. There are 6 generations. So that brings us to times 2 to the 6th. So that is, each of 100 bacteria would produce 64 cells. We had 100 to begin with, so that is 6,400 new cells in our culture. D is the correct answer. So we've talked about the rate at which bacteria grow. However, a population of bacteria does not maintain its potential growth rate endlessly. The population will actually display a predictable pattern called a growth curve, and this growth curve is obviously dependent on the nutrients and such available. The method we use to observe a population growth pattern is by placing a tiny number of cells in a sterile liquid medium. We incubate this culture for over a period of several hours, and then sample the broth at regular intervals during incubation. We're going to plate each sample into solid media and then count the number of colonies present after incubation. So the stages of a normal growth curve are broken into the lag phase, where not much is happening, an exponential growth phase, where we have a lot, a stationary growth phase, where the growth rate has stabilized, and then a death phase. Let's look closely at the lag phase. It's a relatively flat phase. It's newly inoculated cells that have been deposited into our tube. So these cells are going to require a period of adjustment. They need to grow a little or enlarge and then synthesize their proteins and such. So they're growing in size. They're not yet ready to multiply at their maximum rate. The population of cells could also be so sparse that the sampling actually misses them. The length of the lag period really varies from one population to the next. Then we enter the exponential or logarithmic phase. During this phase, the growth curve increases geometrically, and cells reach a maximum rate of cell division. We still have plenty of nutrients, so that's not holding them back. And this will continue as long as there's adequate nutrients and the environment's favorable. But if it gets too hot or too acidic or there's not enough nutrients, then that rate of growth will decline. Next, we move into the stationary growth phase. In the stationary growth phase, the population has really entered a survival mode. They're trying to exist on whatever nutrients are left. So the cells stop growing or they grow really slowly. The rate of cell inhibition or cell death is going to be balanced out by the rate of multiplication. So growth of the population hasn't stopped entirely, but there's not enough nutrient to support all of them. We'll also see excretion of organic acids and other biochemical pollutants into the growth medium that also act to inhibit the growth. After this phase, we enter the death phase. This is where the curve dips down steeply. Cells begin to die at an exponential rate, and so we'll return down to the lag phase. And if the environment becomes favorable again and more nutrients are provided, the cycle will continue. If not, if we're just growing it in a petri dish, then we end up in the death phase and all the nutrients are gone and there's toxicness there, so the cycle ends. So there's some importance in understanding this growth curve. First of all, the implications in microbial control and infection in food microbiology and culture technology. We'll see that antimicrobial agents such as heat and disinfectants can rapidly accelerate that death phase. And microbes in the exponential phase are much more vulnerable to those sorts of techniques. Because microbes in that exponential growth phase are more sensitive to changes, they have higher vulnerability, we're also seeing evidence that growth patterns in microorganisms account for different stages of infection. For example, a person that's shedding bacteria that are in their early stages of infection is most likely to spread it to others because they're in that exponential growth phase. It's also important for us to understand the stages of cells' growth for working with cultures. For example, it's unwise to incubate beyond the stationary phase because additional time is obviously going to cause some decline in population and thus reduce the number of viable cells in your culture. In some applications, a closed batch culturing is inefficient, that is in a petri dish. So instead, we have to use a continuous culture system or a chemostat. This provides a steady stream of new nutrients and removes pollutants, and thus it can stabilize growth rate, so we have continual growth of the culture. Microbiologists have developed several alternative ways of analyzing bacterial growth, both qualitatively and quantitatively. One of the simplest is using turbidometry, which is where researchers are using the turbidity or cloudiness as a measure of how many microbes are growing in the sample. We take a tube of clear nutrient solution, and then as microbes grow in it, it becomes more cloudy. Then we use a spectrophotometer to send light through the sample. If a lot of light passes through the sample, then there's not much growing in it. If less light passes through, then there's more growing in the sample. Greater turbidity means greater population size, and thus we have a qualitative measure of microbial growth. Another method, a more quantitative evaluation, is enumeration method. This method is known as direct or total cell count, and we literally count the number of cells in a sample microscopically. They use a special slide called a cytometer, and that cytometer accepts a tiny sample and it's spread over a grid. So on this grid, we can see how many bacteria are there in that sample draw, and then use it to estimate the total number of cells in a larger sample. Now, there's an inherent inaccuracy in both the turbidity measure and this direct total cell count, because you can't tell the difference between dead and living cells. That's where automated counting comes in. A Coulter counter is an electronic device that scans a culture as it passes through a tiny pipette. As each individual cell passes through the tip, it's counted by the counter. Another means is a flow cytometer. It also measures those cell size as well as differentiating between live and dead cells. A third and more recent technique is called real-time PCR. It's based off of polymerase chain reaction techniques. This technique allows a scientist to quantify bacteria and other microorganisms that are present in our environmental or tissue samples without even isolating or culturing them. So we can take a sample straight from the tissue or environment and measure how many organisms are in there. And that brings us to the end of our chapter on microbial nutrition, ecology, and growth.